Thank you so much for having me, and I'm glad you stayed for so long to finally see my talk. I'm very excited about presenting here. I have a lot of really cool things to show you. Some of the stuff that I'm involved with has to do with the Grower Citizen Science Project, how to build healthier soils. And so I kind of want to change the notion about soils being inert and it's just a medium to grow in. I want to open up your eyes like John Zach did yesterday. Soils are an ecosystem. There are many things going on, and I'm hoping by the end of the symposium you'll have a new perception about soils. And so I'm going to show a little bit about soils, soil structure, a little bit about the microbiology to show you that it is an ecosystem. It's a living entity. First of all, you've probably noticed I have an accent. I am not from West Texas. Surprise. I'm from the Netherlands, and so I'm from a place where Amsterdam, if anybody's been to Amsterdam, yep, that's in the Netherlands. I'm from an area where there's a lot of tulip fields and it's flat and we have windmills. I'm a person, and Kelly knows this as well, I like to see the overlap of things. And so one thing that West Texas has in common with the Netherlands is very flat. So anyways, a little bit more about myself. If you were to think about the papers and the studies that I've been involved with, these are some of the words that come up. So you'll see soil, you'll see quantitative, and carbon, and that's kind of central. And so if you were to organize this, so this is a conceptual diagram as to what I do. I study how the environment affects plant physiology, how the environment affects microbial ecology, but also how plants and microbes interact. In the end, I'm interested in the carbon balance. So to build healthier soil, one key ingredient for that is to build carbon. And I'll tell you why building up the carbon is important. I've also, I've done work in the desert, as you saw on the graph, on the slide before, but also worked in forested ecosystems. So I've worked in a vast range of ecosystems, and a more recent one is Antarctica, so a polar ecosystem. So this is me hiking up on one of the protected islands in Antarctica. And I learned a new skill, how to drive a Zodiac, which is really a lot of fun. One of our study sites was on an island, and it has these amazing plants. One thing about Antarctica is we have no trees, but we do have moss, and we have a grass, and we have a pearlwort. So there's only 2 vascular species. There's no corn, there's no cotton, there's no other plants. It's mostly the bryophytes, which are the mosses and the lichens. I study the carbon flux. So my worry about what's going to happen with the soil. Soils store about 4 times as much carbon as the atmosphere, right? That's a huge reservoir. And what happens to the soil carbon will have an impact on how the climate is going to be changing as well. So here I am with my student Kelly in Antarctica, and we are placing this chamber onto this stainless steel ring to completely seal it. The goal is to figure out what's going to happen with the carbon dioxide, right? Plants use carbon dioxide for photosynthesis. So if photosynthesis is more important in this ecosystem, then the CO2 within here is going to go down because the plants are taking it up. But what about the microbes? Microbes release carbon, right? They decompose, they lose carbon as CO2. So there are soil microbes in here. Who's the winner? Are plants gaining more carbon and putting that in the soil? Are the microbes decomposing it, losing more of that? So this chamber and the measurements I take will tell you a little bit more about whether there's more decomposition relative to photosynthesis. I'm always interested in the carbon balance. And so here's the chamber as a close-up, and we have a little Lego mascot right here, and we also have 2 friendly visitors who came to our site as we're doing this. So this is the neat part about working in Antarctica. But I've done my PhD actually closer to home. I've done my PhD at Big Bend. If you've seen John's talk, you've seen pictures of Big Bend multiple times, and yes, you should go if you haven't been. Big Bend offers really amazing opportunities to see elevational gradients. It's just absolutely stunning. So here I am working on a watering. Yes, we water in the desert. This was one of my experiments, and there's a lot of water in that tank. So as Kelly mentioned, I'm involved in a grower citizen science project. It's really important because there's a lot of concern, right? And this is why we're also having this symposium. I want to help you guys build healthier soils. And for that, I also need your participation. I'll talk a little bit more about the Grower Citizen Science Project to show you also why this is so important. This project that I was leading was picked up by the Smithsonian Institution. And there's actually a video that was taken. So here's a film crew, and that's RN and his daughter. They're in the tractor here. And anyway, they came out to film us. And I want to share with you the video that is currently at a permanent exhibit at the Smithsonian. I don't know about you all, but RN just has a way of speaking this wonderful language and just brings back the message. So thank you, RN, for contributing that and to teach us more about the soil. I also want to acknowledge another group of people, and they are called the Carbon Keepers. So these are a group of elementary school kids, so 6th graders, and they had a really cool science project. So they are worried about the CO2 in the atmosphere. They are worried about what can be done about it. There's no point in having all the CO2 up in the atmosphere if we can use it in the soils, because it helps plants, it helps microbes, it helps everybody. So they designed, or we helped design, and they led the work, and it was their idea as to what are some of the practices that we can make soils healthier. How can we increase carbon in the soil? So a little bit about your project. They have a website as well. And here's some experiments that they have. I'll show you the website in a minute. But yeah, so how to increase carbon and in the soil and basically remove it from the atmosphere. So I have a surprise. They made it here today, so I would like them to stand up and to thank them for the work that they're doing. Thank you. Thank you. Yeah, so it's really, really nice, and it's just so inspiring for me to work with youngsters that want to help growers, they want to help fight climate change, and this is a win-win. And so I mean, that really touched me, and so I hope you feel inspired as well. There is hope. Somebody asked Dr. Zach earlier, and I think the kids are the future, and they are our hope. So why worry about the carbon? Well, first of all, we have lost a lot of carbon already in our systems. Here is a graph that shows you how soil carbon content changed over time. And this is for the central US Corn Belt and a portion of the Great Plains. So early on, we had about, well, I guess 8,000 grams per meter square of carbon, but we only, over the course of the time, around the 1940s and the 1950s, we only had about half of that carbon left. Now that we're going towards reduced tillage, we're starting to rebuild some of that carbon. So why have we lost all that carbon? Well, first of all, here's two images, and they're from the Llano Estacado area. Back in 1541, when Coronado crossed, they called it the Sea of Grass, right? It's pretty productive. And when the Spaniards went through, they basically stated all you could see was buffalo and sky. I mean, it sounds beautiful to me. But you can see there's all this grass, and now this looks very different, right? So just by removing plants, right? That's what we do, we harvest, right? I mean, you can't avoid removing the plants. But one of the main reasons we lost a lot of carbon in the soil is, first of all, this is how the soil looks like in a prairie. A lot of the carbon is actually stored below ground in the root systems. So if you see the image on the left, these are perennial grasses, and these are annual grasses. And you can see there's all this whitish mass of roots, and they extend pretty far down. So most of the biomass, most of the carbon in these ecosystems is below ground, not above ground. The second thing is that not only do you physically have more roots and carbon, those roots also leak carbon. They leak it, we call it exudates. And this is important. Like, why would they lose all that valuable carbon? Well, first of all, it's like your stomach. You have acid in your stomach. These are organic acids that are exuded so that the plants can procure nutrients from the soil. In addition to that, it feeds the microbiome as well. They feed the microbes because microbes will help release more nutrients from organic matter. So they do it for their own benefit. The second part why these exudates are so important is not just fuel for microbes and to do what they do best, right? Nutrient cycling. They also act as glue to hold soil particles together. So having a diverse microbiome is really important. If you get sick or you got a stomach bug, it's probably a weedy species of bacteria that has taken over. What you need is actually a more diverse microflora. It's the same for the soils, and plants can actually influence what microbiome composition they want to have. So the other reason why we lost a lot of carbon, and actually this might be the main reason, is by breaking up the soil through tillage, we're breaking up aggregates. Those aggregates are really important. I don't know if you have till and no-till soils, if you grab a sample of soil, the soil samples from the no-till fields will hold better together. They're more clumpy, right? These aggregates are super important. Once you break it up, so you can see here, this is what a no-till field would have, is these aggregates. They're clumps of soil particles that are combined with roots and hyphae and microbes and all that, and they form these tight nuts or clumps. Once you break that up, a lot of that organic matter suddenly becomes available for decomposition. And then the microbes will decompose, and then you have very little organic matter left. So you're actually exposing organic matter that would otherwise not have been available for microbial decomposition. So having a poor soil structure and having unstable soil aggregates will also result in, when high winds come through, right? So Lubbock area, we get pretty high spring winds. It gets really dusty, and all the dust is actually very fertile soil, right? It's no longer in the land. We want to keep all that fertile soil on the land and not up in the air. And you may have noticed also, if soils don't have good soil structure, that you also have ponding of water and runoff of the water. So aggregates are really important because they have more pore space. Soils with stable aggregates, with stable I mean that they don't break down into smaller bits. If you have stable aggregates, there's these beautiful channels where water can infiltrate. So soils with a good aggregate structure can also hold more water. If you have unstable aggregates, let's say you till the soils quite a bit, those aggregates are not stable and they break down into smaller bits and they start clogging these pores. So rather than the water infiltrating, it tends to run off. You often see some kind of crust forming as well on these soils, which also prevents the water from infiltrating. And you probably have seen something called the slake test. So this is the difference between a soil with good aggregate structure and a soil that doesn't have good aggregate structure. So here's one with good structure. This is some soil suspended in water. And this is a soil with unstable aggregate structure. And you can see that the water tends to get murky. Right? We see that also in a lot of the tilled fields is when it rains, the water is not clear. There's a lot of particles in there. So having a good soil structure is key, and the exudates from the plant will help build some of that structure. And it's not just the plants doing it, it's the microbes as well. When microbes are decomposing the organic matter and they live and die and whatnot, there's these glue-like particles that are being formed, and they help bind those soil particles together. So here is a zoomed in picture. This is it's microscopic. It's a scanning electron microscope image of how one of the aggregates will look like. You can see here a conceptual diagram. It's composed of soil particles, the pore space, the gray is the organic matter. Then if you have soils that have clay, they can form clay mineral complexes. So clays are also very good at creating that. So you can see all of these are involved, right? So there's fungal hyphae and microbes and all of that. And these aggregates are also home to the microbes, right? And so they're important for a good functioning of your soil. So not just for water infiltration, but also to promote microbial activity. So why increase soil carbon? So carbon is very important for a variety of reasons. You need carbon or the soil organic matter to build good soil structure, right? So, and organic matter is also a source of plant nutrients. And so you can see here that increased soil organic matter is the source of plant food, right? So it helps the plants. It improves soil microbial function. It improves the soil structure. We just talked about the aggregates. It also increases water retention. How does it do that? It acts as a sponge, essentially. This is why when you go to a nursery and you want to get some orchids and whatnot, they always add peat or moss or something. It's just because that type of material just, they just hold on to moisture for a lot longer. And then of course, if you have better soil structure, soil aggregates, right, you can also reduce the erosion through wind and water. So you can keep all that fertile topsoil on your field and not having it be lost through water and the air. And also, increased soil organic matter means that some of that CO2 gets into your soil, which is where you want it. So how do we build soil carbon? And this is, I'm going to show some images as well from my Grower Citizen Science project. So we can physically add carbon by adding residue, or keeping the residue. So that's basically directly increasing it, but also because adding residue will also reduce the speed of the water on your land, right? So the water will have more time to infiltrate. And so more water to infiltrate will help plants grow better as well. And so indirectly adding residue will have multiple benefits. We can also increase carbon input through cover crops. Right? If your field is not in use for your regular cash crop, then you can use winter cover crops to start building some of that carbon in your soil, because again, those roots will exude and leak carbon. The roots will leak the carbon into the soil, right? Essentially, the take-home message really is, if you want to build healthier soils, you shouldn't see it very often. So be it through cover crops or residue retention, you shouldn't see it very often. This is how you can build organic matter. We also build organic matter by promoting a diverse microbial community. As I said, it's soil microbiome. It's similar to your gut microbiome, right? You want to have it diverse. So here's a paper that came out, and they were looking at monocultures, so just one crop at a time or one plant species, and what influence a multi-species area what effect that would have on the soil microbiome. The red arrows, it's kind of hard to see, but the red arrows means it's low diversity of the microbes. The green arrows are higher diversity, and they split up the data into bulk soil, so bulk soil, microaggregates, and the large macro aggregates in the soil. But you see that as soon as you have more diversity of plants, you also get more diverse soil microbes, and that is what we need. How do you get more diverse soil microbes? Well, you can do crop rotation, for example, and even just using cover crop, right? So it's not always the same plant in your field. So that will help build a more healthy soil microbiome. Dr. Zach earlier today mentioned about having a better diversity of microbes and not just feeding the ones that are the weedy species, because they are also sometimes associated with the pests, and you don't want that either. Okay, so there are so many benefits of having a more diverse soil microbial community that implies also increased soil fertility, improved aggregate formation, right? Because they have these compounds that they can exude as well and act as glue. And also you have fewer plant diseases. So there's so many different benefits of having a more diverse microbiome. So how to increase soil health? Well, RN stated it so well. To not disturb the soil any more than we have to. And also by taking care of the soil microbiome. So I think a lot of the conventional way of thinking was really you feed the plant directly, you add fertilizer and you feed the plant. But in reality, what we may have to start thinking about is feed the microbiology, and that in turn will feed the plant. So this is how you can build a soil health. Here are some pictures of microbes in the soil. And you can see these structures. They kind of look like very fine roots, but they're hyphae of fungi. Hyphae are just fungal structures, and they can funnel water and nutrients. And also, we have a lot of mycorrhizae in the soil, but every time we till, we break up the hyphae, right? And so the microbe will have to start again. So it'll be nice to have a more diverse microbial community. We can hopefully promote more arborescent mycorrhizae. And the reason why it's so important is essentially those hyphae, what looks like a fine root system, essentially works as an extension of the root system of the plant. Why do the fungi work with the plants? What do they get out of it? And what does the plant get out of it? Well, it's called a symbiosis. The plant does photosynthesis, and that gets converted into sugars. It's energy. So the microbe gets energy from the plant, and in return for that energy, the fungus will mine the soil for nutrients and give that to the plant. And in addition to that, having this more extensive root system with the mycorrhizae also makes the plant more drought tolerant. And we all heard about legumes and what legumes do, right? They fix nitrogen from the atmosphere. But did you know it's not the plants doing it? It's the microbes. The microbes are in these what called nodules. So there are bacteria that are called rhizobium, or rhizobia, and they fix the nitrogen, and then they give that to the plant, and again they get energy in return. So it's a similar way. So we have fungi and bacteria working with the plant. So how to increase soil organic matter? Healthy soils are covered all the time to not disturb the soil any more than we have to, and to promote more diverse microbial communities. In essence, what we're trying to do is mimic nature a little bit more. So this is what leads me to the Grow a Citizen Science project. And everybody has very different management practices, and all we're looking for is what are some of the good ways to increase soil health effectively. So this year I've had 40 fields. We measure microbial biomass, we measure what kind of microbes there are in the soil, we measure carbon in the soil, we measure soil temperature, moisture, nutrients, all of that. It's such a great way to work together. Now this project is funded by Cotton Incorporated, and Cater has believed in this project since the start, so thank you Cater for doing that as well. And also the James Buddy Davidson Charitable Foundation has funded this project. And it is so important because you guys, you know so much about the soils, you know about the system. You know, I mean, I don't know all that stuff that you do, but I have my own expertise, so why not share that? Why not have that symbiosis, just like some of the microbes have with the plants? This is very similar. And if you saw Dr. Zach's talk, he had shown some of these slides as well. We are just worried about higher temperature extremes. We are worried about more droughts as well in this area. So this is a figure that you showed as well. So we're going to be facing some challenges in the future. And the additional challenges, our water source is starting to decline. We are extracting more water out of the aquifer than what is going in. And that is a big concern. So what we're trying to do is, by building up soil health and increasing that carbon and building that soil structure, your fields will have a higher rainwater use efficiency. If it rains, you'll retain more of that water. So what we're trying to do with this project is help you find how we can improve your soil health so that you can face the extremes that are coming in the future. You guys do so much work and you are so important for this region, and I can't emphasize how important it is to make you be successful. And so that's why the Texas Tech Climate Center and our Department of Biology, this is why we're trying to work together with you guys. So again, why is this important? Again, we want to increase its soil organic matter. So what is involved in the Grow Citizen Science Project? Because if you are interested in participating, okay, so these are our little tasks, right? So you guys will collect soil samples just 3 times over the growing season. And then you either drop off or you mail the samples cold, overnight or something, so that we can start analyzing it in the lab. You also, you can measure CO2 for us, where you get this fancy sensor, and you can basically do on a smaller scale what I've done in Antarctica, is measuring whether or not there's a decrease or an increase in CO2. And if you share your yield data, we know whether or not, so we have high organic matter, but what does it mean for the yield, right? So we have more information. And you guys share your wealth of knowledge. Seems pretty easy, right? So what do? Okay, so we'll come out to your field and we'll instrument up soil moisture and temperature sensors, and not just at one depth, but multiple depths. So at 0, so near the surface and then further down. That's kind of where most of the root mass is going to be anyways. We measure moisture and temperature because we want to see if it rains, does it just stay at the surface, or will the moisture actually trickle down towards the roots are seeing it as well. So we'll install this. This is actually Dr. Zach with his graduate student Pablo, and they're still installing this in RN's field. So we also analyze the soil samples for soil nutrients. We examine the microbial community, so we get total microbial biomass and a sense of their diversity. And then we share our findings. You get a report, you learn more about your field. Do you see how this is kind of a win-win situation as well? So this year I had 40 fields across West Texas, but we need many, many more. So I'm going to show you some results. They're hot off the press. I got these figures out last night. These are 2019 data. So, and what I want to show you is that not tilling the soil, so the bar on the left-hand side is conventional tillage, then we have a bar for minimal till, and then a bar with no till. And how does the soil organic matter change when you go from conventional till to no-till. You can see we can start to build soil organic matter in these systems. But you may say, yeah, but I have sandy soil or I have clayey soils. How is that going to differ? Well, so the nice thing about the Grower Citizen Science Project is we have information about your fields. We'll know soil texture. We can start categorizing things. So this is just an example. First thing to note, by the way, is it's very hard for sandy soils to increase organic matter. And those who are dealing with sandy soils probably already know this. So I'm just stating the obvious, but it's nice to see it in data as well. But you can see that irrespective of whether you deal with sandy or clayey soils, there's this upward trend from conventional to no-till to increase carbon. We also have higher soil organic matter from tillage to no-till. We also have increases in organic matter whether or not you have dryland ecosystems or whether you are irrigating your systems. So irrespective of that. So now you can see how I'm starting to categorize things, right? So it's really beneficial to start increasing that soil organic matter, and you can see that not tilling your soil already has a big impact. For the reason that I had stated earlier, it's because now you're starting to build those aggregates. So another benefit as well is that, and I stated this earlier as well, if you are building soil structure, your soils will be able to hold more water as well. So here's the volumetric water content. And again, we have the tillage all the way to no-till on the right-hand side. You can see when you go from conventional to no-till, how you already have higher soil moisture. All right, so we also measure microbial biomass. And there should be a relationship, a healthy relationship between soil organic matter content and how many microbes you have in the soil. Why? It's because organic matter is an energy source for the microbes, right? So there should be a correlation. So you can see that in no-till soils, you have higher organic matter, microbial biomass, whether or not you have dry land or irrigated, compared to the tilled soils. But importantly, what about the microbes? So one of the analysis we use, it's called FAME. It's like my favorite acronym because I used to watch the show FAME a long time ago. It's a fatty acid methyl ester, that's what it stands for. It's basically you looking at the microbial membranes to figure out, okay, is this an arbuscular mycorrhizae? Is this a gram-positive bacteria? So we have these broad categorical variables or broad categorical groups of microbes that we're looking at. And one thing that of course we are interested in, what about the microbes that help the plants specifically, so the AM fungi? So here we have the graph on the arbuscular mycorrhizae, and again I show this graph with a cotton plant and the extended rooting system, you know, thanks to the mycorrhizae. And you can see that in no-till, especially in irrigated soils, that not tilling your soils, you increase that beneficial fungus. And that is because you're not breaking up the soils every time and breaking up the hyphae, so that the AM fungi already has a running start the next field season if you don't till. So this is great. So we're starting to see a lot of benefits of not tilling in terms of soil organic matter, in terms of soil moisture, in terms of the beneficial fungi. We also find that retaining residue has a lot of beneficial impact as well, and that is in part increasing soil organic matter. This is a field, I think this is Jeremy Brown in his field, and he's showing how the soils look like. And it looks like a very nice structured soil. You can see that when you retain more residue in the soil, especially retaining a lot of residue, you can increase the soil organic matter quite a bit. And this is an irrigated field. And retaining residue, here we have So this is the part where I'm missing some fields. There are no fields in my study that are dry land and have very high residue. And I know it's very hard to build up. But if we had a bar here, my expectation is that it would be even higher, right? So adding residue improves your soil organic matter content. And as I said, adding residue also helps slow down the water flow, so it has more time to infiltrate. But for it to infiltrate, you need good soil structure. You can see how things are connected. So the other benefit is, if you have high residue in your field, it's actually been shown that it is also very good for weed control. So those that have a lot of residue in the field also find that they have fewer problems with weeds. So some other preliminary conclusions are that no-till and residue retention do increase soil organic matter, reduce tillage, actually especially no-till, right? So minimal and no-till, they increase soil microbial biomass, including the AM fungi, so the arbuscular mycorrhizae fungi. And also these fields have higher rainwater use efficiency, so more moisture can remain in the soil. So I hope I kind of opened up your eyes, just using John's word from yesterday, to the fact that soils are a living entity. There are a lot of interactions going on. And I hope I gave you a little bit of a sense as to the complexity of soils. And I hope together we can help build soil health to help you guys out. So I found this in on a website from Purdue University, that if you're like, well, I'm not interested in the soil health per se, well, there are also financial incentives to go towards no-till or at least conservation tillage. So that goes from economics, right? Decreased labor time. There's less input in these systems, less wear and tear on the equipment, reduced fuel costs. And then again, the benefits would be increased soil health, and again, higher rainwater use efficiency of these fields. So you don't have to water as much. We can save some on that water, and it saves you money. So, but we need more fields. If anybody wants to participate in my study, I have my email set up here. So if you are interested, please contact me. I think the power of this approach is it is a Grow a Citizen Science project where we all are working together towards the same goal, is to help you guys. It is so nice to have multiple data points. I can really start to get more into details. Everybody does whatever they do with their field. We're not telling you what to do. You just go your own way. We are just monitoring. But by monitoring, we learn what practices are good for certain things. And besides, I really enjoy working with you all. So anyways, I'm a little selfish in that respect. Okay, so in the end, I want to thank not just the growers. I want to thank everybody, also just for being here and for listening and having an opening ear. I just find it very valuable for us to have these meetings. So again, I can't specify to our end how much I appreciate being able to present here, because you guys are awesome. And anyway, we hope we can contribute to helping you guys out. So thank you so much.